Hello, this is part two of our lecture on the history of botanicals, which brings us closer to the modern times. As always, the second slide shown here lists the sources I used in preparing for those lectures. So when we start talking about our modern understanding of botanicals and natural products which make them, we need to talk about the methods which are used to actually determine composition of those products. Hence comes analytical chemistry. And the history of analytical chemistry goes back, at least in Western civilizations, to ancient Greeks. So Empedocles and Aristotle, about 300 years before our era, have suggested that everything on Earth is made out of just four elements. Those are water, earth, fire, and air. And Aristotle then added qualities to those elements like moist, dry, hot, and cold. And remarkably, for almost 2,000 years, this is what everybody believed in. This is what everybody thought is just unquestionable knowledge. And then it started to change. Then science and evidence-based reasoning started to dominate our thinking in probably 17th century. Those things slowly started to change because people started to question. The evidence suggested there is more than just those four elements, and they may be very different. The first term, analytical chemistry, was introduced by Robert Boyle in his 1661 book called The Skeptical Chemist. And then slowly and surely the methodology behind current analytical chemistry techniques started to appear. For example, Frederick Hoffman, again in the 17th century, have introduced the gravimetric analysis. Very important. This is the birth of quantitative science, quantitative chemistry. Gravimetric analysis is a technique for which the composition of compounds can be determined through the measurements of masses. At this time, weighing things became very precise science. So based on the masses of the reagents, we then can understand the composition of the final product. For example, if we start with the exact quantity of metallic iron and then oxidize it with atmospheric oxygen, we form rust, which is ferric and ferrous oxides, a mixture. It is heavier than the original mass of iron we started with, and then we can calculate how much other elements have been added to it by measuring precisely the mass of the reactants and the final product. Stoichiometric analysis, which is also a functional analysis which is based on a conservation of mass, where the mass of reactants equals to the total mass of the product. This is also very important because if we start measuring this, it means that the amount of actual compounds and its composition can be calculated very precisely, knowing what we started with. It's similar to gravimetric method. Antoine Lavoisier, 18th century, was the first one who actually disproved the phlogiston theory and demonstrated the conservation of mass and recognized oxygen and hydrogen, not that fire, which supposedly was the element of all combustion. He realized that actually it requires oxygen to burn many things. Priestley, Cavendish, Lavoisier, and Schily in the 18th century and Bunsen in the 19th century established very important techniques for analyzing gases and the way gases react with each other and occupy spaces, occupy basically vacuum. Spectroscopy is a most important part of analytical chemistry. There are many different spectroscopies which we use now. But the term spectroscopy first was born from flame spectroscopy, analyzing the color of different elements as they burn in the flame. And that was introduced by Talbot, Kirchhoff, and Bunsen burner, remember? That was in the 1840s. And finally, in a really revolutionary insights, in the middle of the 19th century, Frederick August Köchele, and who was German, and Archibald Scott Cooper formulated the theory of chemical bonding. And from that, started drawing actually the structures of the molecules made of elements. No more fire, no more earth, no more water, but elements in the periodic table as we know it now. And actually here you can see the first structure of benzene, correct structure, which was drawn by Köchele in 1865. Well, but let's get back to botanicals and to the history of botanicals. And when in the 17th century, Americas are discovered already. Spanish, Portuguese, and soon British will be flooding its shores. And of course, one of the very important observations which came from this new continent, this new frontier, was the fact that people there are also using botanicals as healing agent. And very soon after Europeans started to come to the Americas, Jesuit priests, for example, learned that Peruvian Indians used the chinchona bark to treat malaria, which was a plague in both New and Old World. They brought some chinchona bark to Europe and truly saved probably millions of lives. In 1820, then isolation of compounds became more common. The French chemists Pierre-Joseph Pelletier and Joseph Benjamin Chevantot isolated actually pure quinine from chinchona bark. And again, used that pure compound to treat malaria. Quinine is a white crystalline alkaloid which inhibits the detoxification of the heme. When Plasmodium malaria is based on the simple protozoan parasite which lives in erythrocytes and feeds on heme and hemoglobin. And heme, which is a part of hemoglobin, is very toxic. So unless it can detoxify it, the Plasmodium will die. And quinine actually blocks that process of Plasmodia deactivating the heme. And that's what kills the parasite. Major life-saving drug was born. In the 1940s, quinine was slowly replaced by synthetic derivatives, which had fewer side effects, were more efficacious, like chloroquine, and there are many of them now. Well, I would like to introduce to you a scientist who probably is not very well known, but yet, to me, he represents one of the greatest minds who ever lived. He's a Scottish physician working for British Royal Navy. His name is James Lind, and his work covered the middle of the 18th century. At that time, of course, Great Britain had the strongest, most powerful navy, was involved in wars and world explorations just like other nations. And the fact which we don't realize very well now, but the truth is that actually through the long sea voyages, navies of all countries would lose more sailors to a terrible disease called scurvy than to military action. For example, during the 7-Year War, there are very few British soldiers which were killed in action, but 100,000 was killed by scurvy. As we know now, scurvy is a deficiency of vitamin C in the body because the rations which sailors would get in the sea, just everything dried, and vitamin C is rather unstable compound. So mainly dried meats, dried fish have very little or no vitamin C in it, and the deficiency in vitamin C would lead to the slow deterioration of the connective tissue. You start losing your teeth, your joints start to deteriorate, and there's incredible pain and aches and fevers associated with that, and slowly you die. And no one knew what causes this terrible affliction. So James Lind decided to figure it out, and the way he approached it resulted in what I at least believe is the first truly scientifically designed clinical trial. He developed 6 different treatments for scurvy in sailors. Cider, garlic, mustard, and horseradish powders, seawater, sulfuric acid, vinegar, and then 2 oranges and lemon. Those were given as supplementation to sailors who actually developed scurvy. What was observed was rather remarkable. Addition of 2 oranges and a lemon to a daily diet of sailors, fresh oranges and lemon, they do contain vitamin C, lead to a complete recovery, complete cure. Partial recovery was obtained with cider, which has little because it's still a processed type drink. Since then, millions of lives has been saved. James Lunt tried to again isolate vitamin C, and there he failed because it's a very unstable compound. But nevertheless, sailors started eating those fresh foods, fresh vegetables, those high in vitamin C berries and fruits, and scurvy basically was totally eliminated as a terrible affliction. 1785, another turning point in the history of botanicals. Another Scottish scientist, William Withering, who graduated from Edinburgh Medical School, publishes a book. It's called Accounts of the Foxglove and Some of its Medical Uses, with practical remarks on the dropsy and some other diseases. What Withering was interested in is treatment of a disease which is now known as congestive heart failure, dropsy at that time. And he heard that digitalis, or foxglove, a common plant in Europe and in England at that time, was able to provide some relief. His contribution to this was that he, again, designed very precise, very specific experiments using foxglove, using different compositions of extracts, using different methods of extracting foxglove, giving it to different patients with congestive heart failure, and taking copious notes on the results and analyzing them with scientific vigor, which only today's science requires. So as a result, at this point, he didn't know what the active was. Now we know it's a digitalis, has a digitoxin, which are this cardiac glycoside molecule which makes your heart beats stronger and can alleviate some symptoms of congestive heart failure. Through his work, through the work of William Withering, the treatment of dropsy became possible, and he formulated the best possible extract of foxglove, which was subsequently used till we isolated the compounds and could make some synthetic analogs. And still today, many drugs for congestive heart failures are based on this cardiac glucosides, which first was studied by William Withering. 1899, the year when modern pharmaceutical industry was born. And its origin lies in a simple plant-derived compound called salicylic acid. As I already mentioned, those papyri from Egypt, Hippocrates in his work in Greco-Roman times, and actually Native Americans living in America, refer to willow bark as a remedy for fevers and pains. Chewing on this would alleviate fever and make you sweat. We know that Lewis and Clark, for example, used willow bark tea as a remedy for fever and took it with them on their famous journey to the Pacific Ocean. In 1838, the Italian chemist Raphael Perea isolated salicylic acid from the willow bark and identified as actually active compound of willow bark, but the name aspirin actually came from a different plant, Spirea ulmaria, which also contain salicylates, different salicylates. So Spirea became aspirin. And the reason we got aspirin is because salicylic acid itself was rather toxic. It causes ulceration, and it's not a very good drug. So a German company, chemical company, subsequently a pharmaceutical company called Bayer, and scientists working there have realized that if you acetylate salicylic acid to form acetyl salicylic acid, you can reduce the undesirable side effects. And this is what became aspirin. And in 1899, aspirin was marketed by Bayer and very soon become one of the most popular drugs in the world. Still is. It's been 120 years. And it's hard for us to realize that the whole pharmaceutical industry is just 120 years old, and yet it has an enormous impact. This is when a synthetic chemistry combined with knowledge of plant natural products made treatment of diseases much more powerful, much more standardized, and much more successful. Well, in general, many natural products which became successful drugs of today were first isolated in the 19th century because of the advancement of analytical and isolation chemistry. 1804, morphine, founding molecule for many analgesics, painkillers of today, was isolated from opium poppy by Frederick Sertiner. 1831, Henrik Main, for the first time, isolates atropine, prepared the crystals of atropine from Atropa belladonna. Subsequently, it was synthesized, because now we use synthetic atropine, which was first synthesized by Richard Willstatter in 1901. And many, many others. Scopolamine, 1880, ephedrine, 85, vinca alkaloids, well, the first powerful plant agents which are still used to fight cancer, isolated in the 1950s, very complex molecules. So the race truly goes on and still goes on into today. In our laboratory, we work a lot with metabolic syndrome and type 2 diabetes and natural products from plants which can prevent and cure those conditions. So therefore, I need to talk about the most successful anti-diabetic drug ever, which also originally came from plant, and this is metformin, used widely throughout the world as cheap and effective medication for type 2 diabetes. It has been known traditionally, historically, that French lilac can actually reduce the content of sugar in your urine, because this is how diabetes was earlier diagnosed. Biguanides are the actives which have been isolated from French lilac as anti-diabetic actives in the beginning of 20th century. Again, somewhat toxic, somewhat ineffective, and this story repeats itself over and over again. Natural product is isolated from plant, it's active, but then it's improved. Its pharmacokinetics and its toxicological profile is improved through synthetic chemistry. So metformin is one of them, but still, it owns its origin. It's a synthetic compound, but it's based on a plant-isolated molecule. Extremely effective today. Well, it's probably impossible to talk about botanicals without mentioning Taxol, which saved thousands of lives as a very effective chemotherapeutic agent used for various cancers. It is a very complex molecule, which is actually a terpenoid, which, as we know now, comes from Pacific yew, Taxus breviola. Two scientists played a key role in isolating Taxol and showing its effectiveness. They are Monroe Wall and Mansukh Wani. Actually, Monroe Wall, who led this effort, studied at Rutgers and got his doctoral degree at Rutgers, and then moved to work for NIH and Research Triangle Institute. They've been working with extract from Pacific yew for a long time and showed, again, its effectiveness against cancer. People didn't believe them. They persisted, and their persistence paid off. We now understand that Taxol works by stabilizing microtubules, and stabilization of microtubules in the cells will block cell divisions. And of course, if you block cell division in cancer cell, you can stop cancer. It is still a very important treatment for cancer. Pacific Yew has many terpenoid-like molecules which are similar to Taxol in their structure and decorated with different groups, and therefore have different efficacies from inactive to very active. So it's very difficult to isolate exactly the molecule of Taxol from Pacific Yew. It takes many, many trees and very scrupulous purification methods. So the way Taxol is made today is called actually a semi-synthetic method. Semi-synthetic method of making Taxol relies on grinding Pacific Yews and isolating the basic scaffold of terpenoid, which you can make by dissociating many side chains, and then taking the scaffold and decorating it as Taxol would look like. So this semi-synthetic method is what now dominates the Taxol production, but also it's been shown that Taxol can be made in plant tissue cultures and the tissue culture of Pacific Yew, also by some fungi, but semi-synthetic method is the one which is used today. Again, the marriage between natural products and synthetic chemistry. Well, people in the United States and of course throughout the world were always interested in buying botanical extracts. However, interestingly, before 1994, it was very difficult in the United States to market botanical extracts because there were no regulations which would cover those products. Foods were strictly regulated by Federal Drug Administration and USDA, which is Department of Agriculture. Of course, pharmaceuticals or drugs were regulated also by FDA, but there's nothing for botanical extracts. They didn't fall into any of those two categories. So in 1994, Senator Orrin Hatch from Utah and Tom Harkins from Iowa introduced a bill in Congress, which is now known Dietary Supplement Health and Education Act, or DUSHE, and DUSHE soon thereafter became the law of the land. That introduced a set of regulations and rules about how you can specifically market dietary supplement based on its safety. Dietary supplements, botanical extracts in the US, are not supposed to prevent or cure a disease. The cure or prevention is not even mentioned. It's only supposed to supplement your diet. Nevertheless, of course, people buy them mainly for disease prevention, cure purposes. But you cannot put that on the label. Anyhow, DUSHE gave birth to what we know now, a $40 billion industry of dietary supplements. A big part of it is botanicals, also vitamins, minerals, a variety of other extracts. And that industry continues to grow today. It's impossible to enter now a supermarket or a drugstore in this country without seeing the arrays of little jars with pills or extracts, and many of them contain those botanicals we've been talking about for the past two lectures. And finally, 2015, another important year for botanicals. This year, the Nobel Prize in Medicine was awarded to a Chinese scientist, Tu Youyou, who was a discoverer of artemisinin, which is now a frontline pharmaceutical used for treatment and prevention of malaria, which saved millions and millions of lives. It took many years to Tu Youyou to isolate the active ingredient of Artemisia annua, artemisinin, as we know it now, and for those efforts over several decades, she was awarded a Nobel Prize for Medicine. And that, again, emphasized the importance and prominence of natural products in treatment and prevention of human diseases. Well, thank you very much for your attention and for staying with me through this long journey through human history, from the dawn of our civilization, when archaeological evidence already showed that early people have used plants for healing till very modern times. We have talked about many important dates, many important peoples, many important events, all related to the intimate connection between our civilization and natural products made by plants, which are not only used for food, but used for health and healing. I am very grateful for you trying to stay with me. This last slide just summarizes those important events, important names. I'm going to keep it on the screen just a little bit longer so you can use it as a summary of the last 2 lectures. Thank you and goodbye.